discuss this thing, that viruses are basically a type of organism uh, which are neither living nor non-living. So why I am saying like that? Why the like I am saying that the viruses are neither living nor non-living? Look, so when there is a characteristic uh, that which define that particular thing is living or non-living, basically, right? So first thing is that whenever these virus they go inside any living body, go inside living body, right? When they go inside any living body, what they do, they multiply, right? They start multiplying. They what they do, they start multiplying, right? They start multiplying. So when they start multiplying, so multiplication is a sort of what? Multiplication is a sort of reproduction, right? It means they are reproducing. Reproducing. Just remember, I don't know. Right. Pen is not behaving well. Give me 1 second. Let me check.
Okay.
Oh, what happened today? Your energy level is low. Are you
No,
exhausted?
no, I'm not. I'm just bored.
Okay. Just give me 1 second. See, this is not updated.
Okay, no problem.
So in last class, we discussed that the viruses are actually connecting link between living and non-living, right? So basically, the basis on which we were talking, or the basis on which we are saying that the particular thing is living or non-living, this is all about the reproduction and the metabolic processes, right? So reproduction and metabolic processes, when viruses are out of the body, that they do not perform like living because they cannot reproduce. Neither they can reproduce nor they can perform any metabolic. So do you have any idea what is the metabolism or what is the metabolic reaction? Affaf, what are the metabolic reactions?
Breakdown of larger molecules into smaller ones?
That is called catabolism.
No?
Yeah,
Um,
that is called catabolism.
make no. Big to small is catabolism.
Yeah, breakdown of the larger molecule into the smaller one, that is called catabolism.
The animalism is from small to big, right? Making bigger things from smaller ones. I
that
don't
is
know, sir.
that is anabolism.
Yeah, I know.
Then what is the catabolism? Metabolism?
Both.
Yeah, both. When we talk inclusive, that when we're talking about the both, then we can say This is the metabolism, right? Look, now it's doing well. Now it's behaving well. So look, so when let me first tell you why we call it the connecting link between living and non-living. Then I'll tell you what is the metabolism. It's very important, and everywhere it is needed. So this concept should be clear. Fine. So when it go inside living body, or rather than living body, we can say when it go inside the cell, go inside the cell. What it do? They multiply, right? They start multiplying. They start multiplying. If they are starting multiplying, it means the reproduction is going on. If they are reproducing, then they are living, right? And when they are out of any living body, they are out of living. They are any non-living surface, right? They are, or rather than they can out of any living cell. Then they do not multiply. They do not multiply. Multiply. They loses the capacity of multiplication, right? They loses capacity. Loses capacity of, when they loses capacity of multiplication, or they do not multiply, they are not reproducing, and no reproduction. When reproduction is not taking place, we can say they are not living. So they are behaving both way, right? Inside the living body, they are living. Outside of the living body, they are non-living, right? So that's why viruses are neither called living nor non-living. So I told you in the last class, the viruses are, they are connecting
Cellular.
link. Sorry,
They are acellular structures.
a cellular structure, and they are connecting link between living and non-living. Link between living and non, right?
Why do they not multiply outside of any body?
I'll tell you. I'll tell you. I'll explain each and everything. Just note it down. Quick.
Okay. Done.
Done. Okay.
Yeah.
So now, first I'll tell you what is the metabolism, or what is the metabolic reaction. Then I'll tell you what is the structure of virus and why they multiply inside the living body, why they're not. Actually, in basic, in layman language, I want to tell you that virus do not have their own machinery to multiply. They require someone else's machinery, right? So virus, that machinery is present inside any living cell, right? So whenever the virus goes inside the cell, they hijack the machinery of that host, right? And with the help of hijacking that machinery, they start multiplying, right? That's how they behave. So virus, they do not have their own machinery like enzyme and all that. They do not have, right? That's why their DNA cannot multiply. They cannot multiply their own DNA, right? Whenever they go inside the living or host body, they hijack their machinery, they hijack their system, and by using their system, hijacking their system, they start multiplying, right? Then this is the reason. Got it, Affaf? I'll
Yes.
explain it in elaborate manner, right?
Okay.
Okay. So what is the metabolic reaction? Suppose that Affaf, uh, so metabolic reaction. So let's assume Affaf, these are chapatis. So chapatis are made up of
Flour,
flour. So what is the chemical component?
glucose, C6H12O6.
No, not glucose.
Sorry, carbs. But carbs are glucose.
Uh, so what is the exact composition of that roti or chapati?
It has water in it and
No.
flour.
So flour, what is the chemical composition of flour?
I
They are made
don't
up of
know.
wheat. Wheat.
Yeah, yes, yes.
What is the chemical composition of wheat? Come on,
Sure, I might be the dumbest person.
starch. Every grain is
It's not
stored.
sugar?
Yeah, every grain. Remember always, every grain, either it's rice, maize, wheat, barley, any grain. So all grain, the content which is present inside the grain, that is the starch. That is a type
Okay.
of carbohydrate or sugar, right?
Yes.
When you eat, then starch, with the help of your enzyme, starch breaks down into glucose. Starch is polymer and glucose is monomer, right? Got it? Got my point? So
Yes.
whenever we digest, right, what we digest with the help of enzyme, our body break down starch into glucose inside our stomach. Then we use the glucose, and it's another story, right? So suppose that I have a chapati, means I have a starch, and I taken a test tube. Inside that test tube, I put an enzyme, and any of the enzyme, like suppose that trypsin or any of the enzyme, right? So don't take particular name of a particular enzyme, just any of it, right? What can say enzyme? So I mix enzyme in test tube. What we will call this reaction? This will be what we will call this reaction?
Well, it breaks down. So it's catabolism.
All right, it's in vitro reaction, in vitro, and this is biochemical reaction, right?
Uh-huh.
Biochemical reaction. We'll call this biochemical reaction when enzyme is taken out from organism, from our body. We'll call it biochemical reaction, right?
Okay.
When this reaction takes place, this is our cell, right? So in vitro means test tube. When this reaction takes place in outer environment, right? In test tube, we call it in vitro, right? And these in vitro reaction are called biochemical reaction, right? We cannot call it metabolic reaction. The same reaction when it is going inside our cell. The same reaction. Enzyme. Same enzyme is present over here. Enzyme. And this is starch. Right? And this is cell. When same reaction this takes place inside any living environment or inside the cell, we call it in vivo or in vivo, whatever you want to pronunciate, you can.
If
Right?
it's in the cell, it's in vivo.
Right. In vivo.
Okay.
In vivo. In vivo reaction always takes, in vivo means within living environment. When any reaction takes place in living environment, in vivo means in living environment. When these reactions take place in living environment, we call it in vivo reaction, right? They are called metabolic reaction. Right? Clear? So all those reactions which are going inside any living organism or inside living cell, they are called metabolic. Clear?
Yes.
Now the metabolic reactions are of two types. Right? Now the metabolic reactions are of two types. First catabolic and then anabolic. In catabolic reaction or catabolism, right, they are destructive reaction. Destructive reaction. What you told me, that was correct. Destructive reaction where the larger molecules are broken down into small molecules, right? For example, digestion. In digestion, the complex molecules are broken down into simpler molecules. So they are destructive reaction, that is catabolic reaction. Anabolic reaction. Anabolic reactions are, that is when they are constructive reaction. They are constructive reaction. In constructive reaction, something is synthesized, right? Synthesis takes place. Synthesis takes place, right? Synthesis takes place. Example, photosynthesis. In photosynthesis, something is formed, means formation of food takes place. That is constructive reaction. That is called anabolic. Is it clear to you? What are the metabolic reaction? What are the catabolic reaction? What are the anabolic reaction? If you want, you can note it down, right?
Okay. Sir, in anabolic synthesis of what takes place? Something
Anything,
or
something,
something particular?
anything. Like
Anything?
synthesis of protein, synthesis of food, right? Synthesis of fat, anything can take place. Look, suppose that take a very practical example. Suppose that there was like a healthy person, he fell down, right? And like his hand is broken, right? Then he'll get the plaster. What will happen when the bone is broken? Between the bone, what will happen? Deposition of the calcium will take place, and the bone will join. That's what happened during the plaster time. Yes. So that deposition of the reaction means something is constructive, right? So that construction process will be called, you'll say the anabolic reaction is going on over there, right? So any constructive reaction which goes on inside the body, that is called anabolic. Like healing of the wound, that is also anabolic reaction, right? But you know, only it does not happen that the catabolic reaction keeps going on or anabolic reaction keeps going on. Both of the reaction moves simultaneously inside the environment, so we call it metabolic reaction.
Okay. I finished it, sir.
Okay, right. So let's start the structure of virus. The second thing which is given in your notebook, that is, now what is the virus I told you. Now the structure of virus, right? So next page. So the virus which we study, the virus which we see, that is something different, right? So let me show you a diagram of a virus, right? Let me show you diagram. So whatever the structure image of the virus, I'll tell you what is that. Video phase virus. Yes, this is the structure of virus. Visible? Affaf?
Yes.
Hello? Okay. So this is the basically structure of virus, right? The virus which you say, this is the virus which you see, you must have seen this diagram everywhere, right? Right? So this
Yes.
virus is bacteriophage. What does it mean? Basically, this is the Latin term. Bacterio means bacteria, right? Bacterio means bacteria. Bacteria, right? Bacterio. So bacterio term reflect bacterio, and phase term means eating. Phase means eating. So those virus which attack bacteria, they are called bacteriophage virus. Got it? You know?
Okay.
Yes.
The bacteria that eats.
Right.
Okay.
Right. You know,
So
it's a
there are
very
two types of viruses, influenza and bacteriophage.
no influenza. Yeah, influenza is also type of virus. Influenza is also a bacteriophage virus.
So like
Right.
there are two types of virus.
No, no, no. There are different type of virus. I'll tell you. You cannot. This is just a example. Right.
Okay.
Take it as example. It's not type. Right.
Okay.
It looks like coronavirus. Right. Right. It's very deep. So I'll tell you each and everything. So assume this is bacteriophage virus. Right. So actually the composition of virus is that. So composition of virus is that. Look, the outer cover of the virus, it is made up of a protein coat, right? A protein coat means a protein covering, right? Every virus, like same the coronavirus, right? The coronavirus look like this one, right? So that is protein coat. This protein coat is made up of, the name of that protein is capsid protein. Got it? The
Okay.
name of the
So
protein is capsid, right?
okay.
And this complete structure is made up of tiles of capsid, tiles of capsid protein, right?
Okay.
These tiles like
So
structure, right? These
the
tiles,
the protein code in every virus is capsid protein.
right? Capsid protein,
Okay.
right? So the virus which have capsule, their protein code is made up of capsid protein, and the capsid protein is made up of the tiles like structure, and these units are called capsomere. Did you got it?
Yes.
Capsomere, capsule. Got it?
Yes.
Okay. So basically, most of the viruses are made up of. So generally, viruses are made up of outer structure of the virus. This is actually ideal diagram. The outer cover of the virus. So first kind of viruses, they are the viruses. They are made up of capsid protein. So their outer cover is capsid protein, and plus inside there may be DNA, right? DNA. So normally the viruses are made up of. So these viruses are called U virus or DNA. They are called DNA virus, or they are called U virus, right? The another kind of, so every virus is made up of two things, protein and genetic material. So let me complete it. So every virus, so I can conclude the viruses are made up of two things. The viruses are made up of two things. What is that thing? A protein, and the another thing is that is genetic material, or rather than genetic material, we can call it nucleic acid, right? So nucleic acid can be of two types. What are nucleic acid? Affaf, tell me.
Nucleic acid. Oh, it has nitrogenous bases.
DNA or RNA, right?
Yeah.
Deoxyribonucleic acid, ribonucleic acid. So nucleic acid can be of two types, right? So
Yeah.
every virus is made up of either protein and nucleic acid. So if the virus have capsid protein outside and inside there is a DNA, we call it DNA virus or U virus. Another thing is that there may be a capsid protein, means capsule, and plus RNA. These kind of the viruses are called retrovirus. Got it?
Retrovirus. Okay.
HIV is a retrovirus. It is made up of RNA, not DNA.
Okay.
Got it? Clear?
Yes.
So look, this is the ideal structure of virus. The outer cover is made up of protein coat. Protein coat is, that is the capsid, right? Capsid is a chemical, and these structure are called capsomere. And inside there is a nucleic acid. So for example, there may be DNA or RNA. Look, this bacteriophage virus, it have DNA, and here this influenza virus, it have what? RNA, right? So this bacteriophage virus, what we call, this is U virus, and this influenza virus is retrovirus. Clear? And these units, they are capsid, they are protein coat. This one, right? This protein
Okay.
coat is called capsid protein, right? And this have tail, right? These are the tentacle or fibers, right? So this is the basic structure of virus. Did you got it?
Yes.
Please, whatever you want, you can note it down.
Done.
Done? Okay. So let me open your textbook. So look up the structure of virus. The same thing is given over here. Capsid, capsomere, right? And one more thing is given. A specific protein antigens are known as virus attachment particle (VAPs), which target protein in the host cell surface membrane. It is how the virus attach to the cell and they infect the cell. What is the VAP? I'll tell you what is the VAP. This is the structure. It is given over here, right? Okay. So virus can be classification of DNA viruses. I told you, right? DNA virus, RNA virus. Then RNA viruses, they are called retrovirus. Retrovirus, a special type of the RNA virus. They have the protein capsid and the lipid envelope inside, and the RNA that is. Okay. One more thing: how the RNA virus reproduce and how the virus actually reproduce, right? Let me tell you. Virus life cycle. A lysogenic pathway and lytic pathway was given over here. Okay, I'll tell you. So types of virus did you got?
Yes.
Okay, let me tell you. There are only two type of the viruses given over there, but actually the viruses are more type. Should I tell you one more type?
Yes. You can. Okay.
So look Affaf. There is a one more kind of the virus, and these virus do not have any genetic material. And those kind of the virus are called prions, right? Prions, right? Prions are made up of only protein. They are made up of only protein. They do not have any genetic material, right? Now, how does the virus work, right? How does the reproduction takes place in the virus? Let me tell you. Let's what happens. So first, I am telling you what is the VAP, right? How does the VAP work? So suppose that this is a bacterial cell.
Sir, what does rayons contain?
Prions, they are made up of just protein.
Just protein.
Yes. Have you listened the name of United States President? His name was slipped from my mind. He is the father of a Star Wars program. Ronald Reagan. Do you know his name?
No.
No?
No.
That's amazing. Ronald Reagan, he's a, do you know a Star Wars program? What do you want to do?
Yeah, I watched a few shows of Star Wars.
No. I don't watch the TV. I don't watch the seasons. Star Wars, that have nothing to do with the Star Wars program.
Oh, okay.
Yeah, this was the.
I was like, why are you asking Star Wars?
No. Actually, what Ronald Reagan want to fit the missiles in satellite. 50% of the satellite, which is the present in the outer space, they belongs to America. The rest for the rest of the world. So in the sky, 50% of the sky has been occupied with the America only, right? So what he want to do, he want to put missiles on this this one on satellites. And during the wartime, what he was planning to do, suppose that they have the war with the China or any other country, any country across the world, China or Iran or Iraq or any country. So what they used to do, they can put the satellite above that country, and they can drop the that missiles over that country, and within a minute they were targeting to finish the country. So that was the dangerous program of the Star Wars program. And the father of the Star Wars program was Ronald Reagan. He was a very dangerous person and very well determined person, Ronald Reagan. And still America is running that program with the name of the NMD, National Missile Defense System, right? So they are running still that program. So actually the Ronald Reagan, when he became the old, he was very prominent and very famous United States president, and they are the people who made the United States powerful. So when Ronald Reagan was at his old stage, he forget each and everything. What happened? He was attacked by a virus. He was attacked by a viral disease, and the name of the disease is Alzheimer. Do you know the Alzheimer?
Yes.
In Alzheimer, people start losing their memory, and gradually they die. The death is the ultimate thing, right? Ultimate thing, all right? Means to save that. It's a really very Herculean task to save that person if he or she is hit by the Alzheimer virus, right? So because it eat up the nervous system, it damages the nervous system. The
Oh, that's so
same,
bad.
yeah. So actually, the same disease, it hit to the Ronald Reagan. Ronald Reagan died because of the Alzheimer disease, and the Alzheimer virus is a prion virus. This is the
Okay.
end of the story. Got it?
Okay.
Right?
So there's no cure for Alzheimer's?
You can stop that because the nervous system cannot regenerate. You can stop the degeneration at that point, but you cannot regenerate. These are the only cells which stop divide when we take birth, right? They cannot, neither they can regenerate nor they can divide.
So like stem cells does not work on that?
No, no way, no way.
Okay.
You cannot regenerate the nervous system, right? If we would have been able to regenerate the nervous system, then you can like you must have the like then the world will have the cure for the polio disease. What
Oh,
happens in polio? The nervous system degenerate, right? So did you got the Ronald Reagan? Listen, his
yes.
name, Star Wars program.
Yes.
Okay, fine. So this is the bacterial cell. This is bacteria. Do you listen news or not? News.
Sometimes.
Sometimes. Always listen news and word like international news, right? That will help you a
So
lot.
exhausting. That's why I don't watch it. Mentally draining.
Ah, I'm listening news at since the age of the 7 years, when I was 7 year old. This is virus. Now how the virus recognizes that I have to attack on this cell, you know. So virus have a special kind of the protein, right? Where at their outer surface. This protein is called what? VAP. Got it? What is VAP?
Yes.
Okay. These VAP protein, they recognizes the protein present on the surface of the cell where they have, which is the target cell. So they have particular kind of the target protein, right? So this VAP recognizes this protein, right, and attack this first step. So I think VAP must be clear to you, right? Now, what is the virus composition? This outer part of the virus, it is made up of what? Protein. And inside this protein, so that is the capsid, right? Capsid protein. And inside the protein, there is a genetic material.
Sir, what's the full form of WAP?
Viral identification protein. This is given over here. After the class, you have to go through with your this one
Textbook.
textbook, right? So that is viral attachment protein, VAP, right? So this help to attach on the surface of cell. Clear?
Yes.
This viral attachment protein, identify the surface protein. This is surface protein. Let me write it. Surface receptor protein, right? Now what it will do? It cannot divide out, right? Till here, it's clear? Affaf, clear?
Yes.
Now what is the next step? What the virus will do? Virus will work like syringe, right? The next step. It work like injection, right? It's neck. So neck of the virus work like injection. What it will do? This neck part, this neck part will compress, right? This neck part, it will compress, right? Like syringe, whenever doctor gives us injection, that part compress, and it will make a hole over here, right, on the surface of bacterial cell wall. It will make hole. With the help of the hole, what it will do? It will insert its genetic material, right? So this genetic material belongs to virus, right? So virus injected its genetic material. So this is viral genetic. Viral genetic material. The protein part remains outside of the cell. It does not go as a whole inside body. This protein part which falls down out of that bacterial cell, this will be called ghost, right? And this is a bacterial cell. What it is? It is host, right? This is bacterial cell. The same thing it do with our cells. This is called host, right? So host remain outside and host goes inside. What happens? Bacteria also have its own genetic material. You have already told me the last class. So this is bacterial genetic material, which is single circular DNA, right? This is bacterial DNA. Bacterial DNA. Now this bacteria also have its enzyme which help in replication of the DNA, right? Now what it will do again? Now in the next step, now the ghost will fall down outside. Now what it again do? In the next step, it will cut down the bacterial DNA, right? It will make a cut in bacterial DNA, and then it will hijack the bacterial DNA and incorporate itself, right? So next step, what it did, it hijacked bacterial DNA. Now it will ask bacterial DNA to make its own copy. Now the bacterial DNA, it will hijack bacterial DNA. It will use bacterial machinery, means it will use bacterial enzyme. That's why virus cannot replicate outside, because for the DNA replication, for making more DNA copy, it do not have any machinery. It uses the host machinery. That's why it cannot replicate outside of the body, right? With the help of that, it will make multiple copy of DNA, right? Multiple DNA copy or genetic material copy, right? In the same way, it will ask bacterial cell to make its cover, capsid. Now, again, it with the help of it will ask bacterial cell to make its cover, right? It will make capsid or capsule. Now, what will happen inside? Now, the next step, they will start assembly, right? Now what they will do in the next step? In the next step, they will start assembly in the bacterial cell, inside the bacterial cell. Inside the bacterial cell, they will assemble, and when the number of the virus will become 200 to 300, they will rupture, they will kill bacteria and rupture the bacterial cell, right? When their number will become, assembly will take in place. Assembly. Then what will they will do? They will rupture the bacterial cell, rupture, and their number they will multiply them. That's how the virus complete their life cycle. Now they will go out of the bacteria. They will infect another cell, new cells, right?
How does it make multiple DNA cap or cup?
Multiple DNA copy, right?
Yeah, yeah.
They have enzyme. That enzyme is the enzyme which multiply with the help of DNA. What we call that enzyme? Which help in the replication? Come on, DNA polymerase.
Wait. I do remember DNA helicase? No,
DNA
polymerase.
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 Yes. Helicase open the helix, right? Topoisomerases unwind, right? SSB protein provide reassociation. Then DNA polymerase makes new DNA on the template of old DNA, right? It helps the DNA polymerase of bacteria. With the help of DNA polymerase of the bacteria, it makes its own copy. That's it. Then
Okay.
it starts translation. With the help of translation, it makes capsid protein. Capsid protein assemble and makes capsule. Got it?
Okay, so capsid is made when these proteins
DNA
join
start
together.
yes, this DNA start translation.
Okay, so it breaks down itself and goes inside the bacteria and then it assembles.
Right. That's how it
Fe-
completes.
uh, Feeding off the genetic material inside the bacteria.
Right.
Okay.
It cuts down the bacterial material. It's cut down the bacterial material and joined its own bacterial material, join its own DNA with the bacterial DNA.
Okay.
Because without that process, it cannot replicate. So
Okay.
it hijack the bacterial genetic material. Got all the points, Affaf?
Yes, but can you just explain the last part again?
Last part, this
No,
one,
the multiple DNA part.
multiple DNA part. Look,
Yeah.
now it have associated. Now it will what it will do. Now this will start replication. It will not allow the bacterial DNA to replicate. It will allow its own DNA to replicate. For replication, it do not DNA polymerase. It uses the bacterial DNA polymerase. With the help of bacterial DNA polymerase, it make its multiple copy. That's it.
Okay. So what
Like
does
the
it do
art.
with its multiple copies?
Sorry.
What does it do with its
Now these
multiple
multiple
copies?
copy will go inside this capsid.
Okay.
First, inside this cell, one virus will produce 200 to 300 virus.
Okay.
To produce more 200, like 200 to 300 virus, it needed 200 to 300 DNA copy.
Okay.
It needed 200 to 300 DNA capsules,
Okay.
viral capsules. So first it will make 200 DNA copy, then it will make 200 capsule copy. Now the both will associate. Now the new 200 virus will arise. Then it will
Okay. So
rupture.
there's not just one. It makes multiple of it. Its
It
kind.
makes 200 to 300 new viruses. Single
Oh,
virus
okay.
makes 200 to 300 new viruses.
So
Then it kills
the
the cell,
genetic material of this virus
right?
mixes with the genetic material of bacteria.
Right, to replicate, for only
Yeah, it
replication.
mixes or it feeds off the bacterial
No,
one
it not
to
feast,
make it
feeds on. It make a cut and join itself in between.
okay. So it eats that and it makes same
It
copy
cut
of
that.
itself.
It cut that with the help of helicase. It cut.
Cut the bacterial
Look. Let me
DNA.
explain. Let me elaborate it. Look. First, what happened?
Okay.
This is the This is the bacterial DNA. Bacterial DNA, right?
Okay.
And this is viral DNA. First,
Okay.
what viral DNA will do? Viral DNA will make cut. with the help of helices. Now, why? Because it without making cut, it cannot rejoin. So now the space is created over here, right? With the help in this space, it will combine, right? With the help of ligase, it will join. Now it will start replication. Who? Viral DNA. Now the viral DNA will start replication. Now it have hijacked the bacterial DNA, and it will ask bacterial DNA that you have to do what I want. Got it?
Okay. So it joins and then it makes same copies of itself, multiple of them.
Right.
Okay.
And with the help of bacterial DNA polymerase.
Okay.
What is the actual function of bacterial DNA polymerase? To make bacterial DNA copy.
Okay.
But now the virus will not allow that DNA polymerase to make bacterial DNA copy. Now it will ask DNA polymerase to make its own copy.
Okay.
Got it? That's how the
Yes.
replication takes place. Crystal clear?
Yes.
Now let me open your textbook. So the same thing is given here over here, the structure, types of virus and all that. Look. The life cycle of virus is of two types. So lysogenic and lytic. Now I'll explain. Many of the virus do not cause the disease when they first go inside the host cell. They insert the DNA into host DNA so that it replicates. I told you that thing, right? Every time the host cell divides, the inserted DNA is called provirus, right? Clear? That inserted DNA part will be called provirus. Messenger RNA is produced from the viral DNA because of the viral gene causes repressor protein to be produced, right? To repress the rest of the activities, to make impossible to translate the rest of the viral genetic material. The virus is said to be latent during this period, means virus will remain active during this period. This is called lysogeny, right? This period is called lysogeny. When the virus is a part of reproducing host cell, under certain condition, for example, when the host is damaged, the virus in its lysogenic stage are activated, right? Means what happened whenever the provirus goes inside the bacteria or any other cell, it makes some repressor protein. What is the function of repressor protein? To repress the virus, right? But when under certain condition, the when host cell is damaged, then virus, lysogenic virus means inactive virus, it becomes activated, and the amount of repressor protein decreases. The amount of repressor protein decreases, and the virus entered into lytic pathway become the virulent. Did you got this point or not? Affaf?
What's lysogeny?
Lysogeny means the virus remains dormant, inactive state.
So it doesn't do anything?
No, it remains sleeping. Why? Because there is a person who is stronger than that, and it makes if virus will do anything, then it will start beating. That is the repressor protein. If virus will wake up and do any bad activity, then a repressor protein will start beating it. So until unless repressor protein is there, virus will keep on sleeping. But on certain condition, when the repressor protein will decrease, amount of repressor protein will decrease, then the virus will become active. Clear?
No.
No?
No.
Okay.
Okay,
So
so it
let me,
it uses a repressor protein
no,
to stop it.
let me explain. Let, Look, so what I was telling you, that is the lysogenic pathway, lysogenic cycle. Look, Affaf. Bacterial DNA, viral DNA, right? When this DNA will go to defend, then bacteria is not a host. Bacteria don't want to entertain virus. Bacteria will do someone, right? When someone comes
Okay.
to your home and that is not favorable host, then guest, then what you will do? You will offend that, right? You will retaliate with that. So bacteria also retaliate. Bacteria produces a protein which suppress this viral DNA, and the name of that protein is called what? Repression. Got it? So
Okay.
this is a repressor protein. What repressor protein will do? It will repress or suppress the DNA. Now this stays, right? Now this stage will be called what is lysogeny, right? So at this stage, the virus, what is virus is doing? At this stage, the virus is inactive, right? So inactive state of virus is called inactive virus state is called latent stage. So at this time, is virus is doing nothing. It's sleeping. Why it is sleeping? Because of repressor protein. Where does repressor protein came? It came from the bacteria, right? Then this state is called latent state. Why it is sleeping? Why it is latent? Because of repressor protein. So this life cycle, this stage is called lysogen. So in lysogenic time period, lysogenic state.
When does repressor protein come out?
Sorry.
When does repressor protein starts to inactivate?
Where so suppose that the bacteria is not getting the nutrition. Bacteria
Okay.
is itself weak. So when bacteria will become weak, when disease attack us, tell me, when our immune system stop working, when disease attack a human, tell me.
Okay. So that's when the repressor protein comes?
Exactly.
Okay.
So if
So it
repression
comes
protein,
after the multiple copies are made
no, after the
or
viral attack,
before?
whenever any
Okay, so it
unwanted
okay.
guest will come, whenever unwanted guest will come, the repressor protein will form. In the same way, whenever unwanted material or unwanted guest like bacteria or virus comes in our body, what happens? Antibody production start only on that time. Clear? Affaf?
But then how does the virus make multiple copies when there's a repressor protein?
When repressor protein is there, their virus will not make its multiple copy. It will make only when repressor protein is not there.
Okay.
Right?
So in what case is repressor protein not there?
When bacteria is weak, they do
Okay.
not have nutrition, conditions
Okay.
are unfavorable for bacteria. Clear? So what will happen?
Yes.
When repressor protein is not there, then it will start multiple copy. When it will start multiple copy, it will make number of bacteria, it will rupture the cell, and that is called lytic phase.
Okay. So lysogenic was when repressor protein comes out and makes
Right.
the virus fall asleep.
Right.
And then when which one is lytic?
Lytic, let me tell you. Now
Okay.
comes the lytic. Sometimes the viral genetic material replicated independently of the host DNA immediately after it entered the host. Entered the host, it do not require to associate with bacterial DNA. It start replicating itself by using bacterial like AIDS, HIV. It start replicating itself without associating bacterial DNA. The mature virus are made eventually in the host bust. Number of the virus are produced inside the host, and the cell bust, releasing the large number of the new viruses particle to invade the other cell. The virus, now disease causing and process of replicating and killing the cells, is known as lytic pathway. They are dangerous viruses. Lysogenic viruses are less dangerous. Lytic viruses are very dangerous. They enter inside. Their DNA enter inside. Their DNA do not need to associate with the bacterial DNA. It make its own copy. After making its own copy, it rupture the cell, right? When the cell rupture, organism die, and now they are ready to invade new cells.
Okay, so it doesn't attach itself to the DNA.
Right. It do not
And
require.
it just multiplies inside the cell. It just needs to get inside the cell to multiply.
Exactly.
And it multiplies and it leaves.
Right.
And it causes diseases.
Right.
Okay.
Now, what happened with the retroviruses? Retrovirus do not have DNA. Then how they will multiply? So retrovirus, they will work like the same way DNA virus do. Retrovirus goes inside the cell. First, they will make DNA from RNA, because they are RNA virus. They will make DNA. So from RNA to DNA, when making RNA to DNA, they require enzyme called reverse transcriptase. Remember that enzyme?
Repressor?
No, not reverse.
Which one?
Retrovirus. I'm saying, suppose that a retrovirus is attacking to cell. This is a cell. It is attacked by retrovirus, means RNA virus. Right? This cell is attacked by a virus. Now, this virus have RNA. It do not have DNA. It have RNA. So this is RNA. Now, first, what will happen? This RNA will make its own DNA. RNA require an enzyme called reverse transcriptase. Reverse transcriptase. With the help of this reverse transcriptase, RNA will make DNA. Now, this DNA will start replication, making multiple copies. Replication. So for replication means from DNA to DNA formation. Now, number of the multiple DNA will copy will be produced. For it, it will require DNA polymerase. Right. The rest is the same, right? So, but in case of the retrovirus, it require reverse transcriptase, like HIV. Clear?
So where does it get the reverse transcriptase?
They have their own reverse transcriptase with the DNA. HIV virus have reverse transcriptase. Sometimes they take it from the bacterial cell, but normally virus have two things. Virus have its own this structure, their own genetic material, and their own reverse transcriptase.
Okay.
Got it? Is it clear?
Yes.
Okay. Viral disease. What is that? Certain disease. Viruses causes a symptom of the disease. The lysis, the host causes circulation. Toxin inhibit metabolism. Nothing very important. It's example. You have to read it out, right? Viruses
Okay.
and disease. How does the cause disease? You know that how does the virus causes disease? Uh, Bacteria, growth of bacterial colony. Do you know how does the bacteria reproduce? Binary fission.
Yes.
They multiply, right? First multiplication, and the multiplication takes place starts from the where? Mesosomes, right? Multiplication
Yes.
starts from the mesosomes only. From the mesosome, they start dividing, then their DNA divide, then one bacterial cell become two. It becomes binary fission. The time between the cell division is known as generation time. Yes. Growth of bacterial colony, they just take the nutrition. Like suppose that you have a milk, put one drop of the curd, right? That lactobacillus bacteria will multiply, right? They take the nutrition from lactose sugar, and they'll just keep on multiplying, keep on multiplying. That's how the growth takes place. Bacteria, they multiply eventually because every time they become double, right? So this kind of the curve will be formed. Log table is given, but that is not required for you.
When is the second table is formed?
This one,
Yeah.
uh, this is actually for the like those bacteria which do not multiply like in the faster pace, like 2 to 4, 4 to so that's how it takes place. But it is not work. It do not work in the case of bacteria. In bacteria, this exponential growth curve work. This one,
Okay. So it's slow, but it just
right?
gets fast all of a sudden.
Exactly. So lag phase. This is the growth curve actually, right? So, but it do not analyzing the data. You will have measure the growth of the bacterial colony in number of the different ways. When the number of the cells of bacteria culture measured over the time, the pattern called curve. You have to remember this thing. Lag phase when it is multiplying very slowly. Log phase, where log phase exponential phase when the rate of the bacterial reproduction is very fast. Stationary phase when the total growth rate is zero, as the number of the new colonies formed by new fission are equal to the number of the cell dying, right? When the death and the birth is equal, right? Then death or decline phase when the number of the bacteria cell will start dying, right? So
Okay.
this is just a phase of the graph, right? It's nothing great over here. So here you can say if the bacterial cells, the number of the bacterial cells are like mm, dying more, sorry, like the number of new generation are more, new bacterial cell are more, and but some of them are dying. So this graph will form. But normally bacterial cells do not die. Why? Because when they mature, they multiply.
So will we do the calculations?
No, not at all. Generally,
Why?
because it is not needed actually. This is just formula. Look, they
Oh, I
have
just need
taken
to
this.
learn it?
No, you need not to learn that. This is just example. I have
Okay.
never I have never got question in till my PhD that you have to calculate the number of the bacterial cells.
Okay.
That is not
Well, I
needed.
hope I don't get that.
But you have to remember this graph. This graph. It is
Yeah.
important. They
Okay.
will ask definitely this one and lag phase. The same graph is used for the plant as well. Everywhere the plant. It also work in the botany as well. This is exponential growth curve. You can learn this formula. This form,
Okay.
right? Learn this formula, right? What is K? What is they can ask this formula to write it down. Nothing. Not in the form of question. They can write it. Ask you to write it down. Right? Microbial techniques. Okay, this may be important. So let me read it out. What is that? What is the microbial technique? Most of the microorganisms are small, but they are impossible to see with the naked eye. We know to investigate microorganism. The example, to diagnose the disease or scientific experiment, you need to culture them. Culture them means we need to help them to multiply, right? This involves the growing large number of the microorganism. They can be measured and so way. They need to be provided with the right amount of the nutrient and oxygen, and then ideal pH, temperature, and the growth. The bacteria and fungi are the most commonly cultured organism, right? Basically, to culture, we use the either milk or the that coconut water. Coconut water is the best nutrient material to grow the bacterial colony. Normally, we take them.
Coconut water?
Right. That is the most
Okay.
nutrient thing, and that is the most perfect nutrient. That is the perfect nutrient to grow the bacterial colony. Normally, that is used by scientists. Uh,
Okay.
when you grow a pure strain of the microorganism, the entry of any other microorganism from the air or skin into the culture will contaminate. It is important to take great care of culturing microorganisms. So whenever we grow the bacteria, we keep it in that sterile medium where other microorganisms like other virus, other kind of the bacteria, other kind of the fungi do not interest, right? Even if the microorganism you are planning to culture is completely harmless, there is always a risk of the mutant strain developing uh, developing that may be pathogenic. Yes, there is quite enough chances that we are using the harmless virus, we are growing the harmless bacteria, but due to the mutation, there may be chances that mutant means that the harmful bacteria may generate in between them. There is a risk of contaminating of culture by pathogenic microorganism from the environment, like from the air virus can come, so we have to pass the filtered air. So we need that kind of the chamber to grow the bacteria where the air comes after the filtration. Health and the safety precautions must always be followed very carefully when handling, culturing, or disposing the microorganism. All the equipment must be sterile. Sterile means germ free before you started the culture. It is particularly important that the culture you have grown does not have no, does not leave the lab, right? That culture always remains in the lab. You should dispose the cultural safety by sealing them in the plastic bag, sterilizing them in 121 degrees centigrade for 15 minutes under the high pressure before throwing them away. There are no ethical issues associated with the culturing of microorganisms from the respective microorganisms themselves. However, you should always consider the danger of infecting other people with the pathogen by accident, right? Got it?
Yes.
Now, what is the aseptic culture technique, right? Aseptic means non-pathogenic. Culturing the microorganism involves the number of the steps. First, you need to decide which microorganism you want to culture and obtain the culture them. Then you need to provide the microorganism with the right nutrient in order to grow. Most microorganisms require a good source of carbon and nitrogen as well as specific minerals. You need to use nutrient medium in form of nutrient growth, where the nutrient are in liquid form for a liquid culture in flask or test tube in the solid form. Usually nutrient agar. Agar is a type of, what is agar? Affaf? What is agar?
I've seen it. It's like jelly.
Yeah, it's like jelly, right? It's like jelly. So agar is a type of carbohydrate, right? We take it out from the algae, right? Agar, and that's used anywhere, So that is the nutrient medium to grow the bacteria. Agar is jelly extracted from the seaweed. It is very useful because the solidified jelly at 50 degrees centigrade. it does not melt again until heated 90 degrees centigrade. Both solid and liquid media must be kept sterile until ready for use. You learn the basic principle of culturing the bacteria aseptic condition on the agar plates. Some microorganisms will grow on the pure agar, but most but most need added nutrient. The majority of microorganisms grow on or in medium enriched with the good protein source such as blood, yeast extract, meat extract. Some need very precise the balanced nutrient. By producing the nutrient medium with very specific ingredients, you provided a selective medium, a medium in which only selected group of the microorganism with those particularly requirement will grow. Selective media are important in identifying particular mutant strain of the microorganism and antibiotic resistance. For example, YM media have low pH with which encourages the growth of the fungi and molds but discourages the bacteria, while the MAC conky agar is designed to grow the gram negative bacteria. Selective, so these are called selective media because they are required or they are restricted to particular or selected kind of the strains, right? Selective media are also useful to identifying the microorganisms that have been genetically modified. With the scientists, genetically modified microorganisms add a new successful gene. They are they often also added gene for the requirement of a particular nutrient or antibiotic resistance. This act as marker gene. Do you know the marker gene?
No.
I'll tell you the marker gene. Actually, biotechnology in our syllabus will study. It requires time actually. Now the practical skill is also given over there. Now you have to study practical skill by yourself, right? So till this point you have to cover. Okay. Okay, Affaf.
Yes.
So you have to study it. There is nothing. Oh,
Till measuring this new topic.
yes, till
Okay.
here. You have to till 90, page number 98, right? So till this
Okay.
point, you have to read out each and everything. You have to understand. And in case if you are facing any problem, so what you have to do is jot down those points, jot down those questions, right? Then we'll discuss that in next class. Clear?
Yes.
Okay, Affaf. So everything clear, or do you have any ambiguity?
No, it's clear.
Okay. So see you in next class. Okay. This textbook is, I don't know, like how it designed.
It's not very helpful.
It's not very helpful. Yes. And it's not even interesting. But I have gone
Yeah.
through with a number of not only Indian authors. I have number of the books on genetics. Like I have 10 or 12 book on the genetics only, genes only, right? And they are from different authors. Some of them are like Korean authors. Some of them are American author or some other country belongs to other country. But they are interesting. But this textbook, I don't know what kind of the textbook they have designed. What exactly they want to convey this to a
There
student.
are other boards for A levels, and according to my teachers, they are more harder than this.
Okay,
So this is like the most helpful one.
this one.
Yet it, yeah, yet it is very insane.
Mhm. Okay, fine. No issue. Whatever we have to read out that, right? So complete it till this point.